How do we measure anything in the body? And how do we analyze and understand these measures? How can we tell when someone's blood pressure is high, when their breathing is uneven, when their brain is malfunctioning? All of these are variables of which the measure and analysis is literally a life or death matter. There is always a device that does the job, but how do these devices work? Here is the path we take in order to quantify our meaningful parameter and get relevant information out of it. The variable is first detected with a bioreceptor. The bioreceptor can be DNA sequence, which is complementary to the DNA we are trying to find and measure. Antibody, which recognizes a specific type of molecules, binds to them and therefore highlights them. Enzymes, which help reactions happen in the presence of certain reactants and therefore changes the chemical combination of the solution where the change is detectable, like a change in the pH value. Cells are biosensors, which are used mainly to indicate cell-cell contact and setups to determine metabolic products. Nerve cells also are able to detect neuroactive compounds. This change is then fed into the transducer, the job of which is to transform one type of energy to another, and thus it will output a voltage signal if needed. However, this voltage signal is impure, affected by noise and measuring effects, and is not ready to be analyzed. Therefore, an analog-digital converter is used in order to purify the signal and shape it into the best form needed for our final step. The signal is then displayed and stored, available to be read and interpreted. Its interpretation happens through signal processing, which includes a lot of features for the processor to choose from, depending on the application. An example of this chain would be to measure the brain activity through EEG, in which case we use electrodes to measure the electric field outside the skull. After amplifying and purifying the signal, we can watch the behavior on our computer and are then able to tell if the person is asleep, awake, thinking and concentrating, and to extract other information. One way to form potentiometric transducer is to have an ion-sensitive field effect transistor, also known as ISFET. The ISFET is composed of a p-type substrate, which includes electron and electron holes, while the two regions above are composed of an n-type doped silicon, which includes free electrons capable of moving around. This is called a p-n junction. An insulator is placed on top of it. The regions are now divided into the source, the gate, the drain, and the bulk. The objective is to control the flow of current between the source and the drain. If this is all that there is in the figure, the source will not be able to send any current to the drain because of the separation between the two n-type regions. However, if we apply a small positive charge on the gate, the electron holes will repel and the electrons will stay on the top area. The more we apply positive charge on the gate, the more electrons are present in this area. At a certain threshold, the current coming from the source will finally be able to pass because electrons will be carried from the first n-type region to the next. This particular type of sensor has been used in neuroprosthesis for many reasons. When the neuron fires, ions go in and out of the neuron, changing the charge of the medium on its outside. If an ISFET is connected to this extracellular medium, the change in the charge will be detected, will change the current flow, and the neural activity can thus be recorded. Frequency coherence plays a role in measuring how much two signals have similar frequencies in order to understand where these frequencies are being used. Frequency spectrogram allows us to see which frequencies are being used in time. For example, how much does the frequency in my brain differ if I hear someone calling my name, turn around and see them, or if I see someone coming towards me and then they call my name? There are a lot of nice discoveries found because of frequency coherences. Kumagai et al. Showed high-frequency coherences between signals coming from listening to familiar music versus unfamiliar music, and they concluded that this is probably why we enjoy the same music so many times.